Welcome to video 3, Lock and Key Hypothesis. The lock and key hypothesis is used to explain mode of action of enzyme, or simply how enzyme works. So I'm very sure that most of you are familiar with how a key works. So the key will fit into the lock, and the shape of the key must be specific to the lock in order to open the lock. As you can see here, the golden key is not able to open the lock, but the silver key is able to open the lock. And that's because the shape of the silver key fits perfectly into the lock. So, using this model, the enzyme is like the lock, and the substrate is like the key. Let's look at how this will match the mode of action of enzyme. So the substrate over here will fit perfectly into the active site of the enzyme or the lock to form a complementary fit. How about other substrate? Will it be able to fit into the active site of the lock? Of course not. That's because the shape of other substrate is not complementary to the shape of the active site of the enzyme or the lock. So there's no complementary fit, hence there's no reaction. So let's now do a quick check. Amylase is an enzyme that breaks down starch into maltose sugar. Use the idea of lock and key hypothesis to explain why amylase is not able to break down protein. So let's look at this question and sieve out or identify all the keywords that will help you answer this question. So it says here that amylase is an enzyme. So here you must already be able to make the connection that amylase is the lock and that starch is the key. So why is it that amylase is not able to break down protein? Since you need to use the idea of lock and key, you must be able to connect the ideas together. So amylase is the lock and starch is the key that will be able to unlock. Protein is then not the correct key with the wrong shape. So this means that protein cannot fit into the active site of amylase or the lock. Right, so now how do we write it in sentence? So we will start with saying amylase is the lock and that starch is the key. And protein cannot fit into the active site of the amylase. Since there is no complementary fit between the protein and the active site of amylase, protein cannot be broken down by amylase. So whenever you encounter questions like this, you can always use this format to answer the question. We've come to the end of the video. Goodbye.